Great. So we can start. So, hi everyone. Thank you for joining in. If you can turn on your video, that'd be great. Professor Udin can have a look at your faces rather than staring into the oblivion here. So, a brief bio about Professor Udin. Again, I met her during a very lovely, very intimate conference in Washington University, St. Louis, and I had known about her work for a long time. Again, I read a record of a few papers before then. It was lovely meeting her, and again, we have collaborated on a paper and we are continuing that collaboration. And she does very amazing work. And very interestingly, we did have a conference here where we listened to Professor Menon in
Yeah, yeah.
and he did talk about very interesting work and a lot of papers that you and him have worked on. So it was again lovely, kind of bridging all those gaps together. Um, so, brief bio about Professor Rudin. She received her PhD in cognitive neuroscience from the psychology department in UCLA. She completed a postdoc at Child Study Center at NYU. She has worked as a faculty member in psychiatry and behavioral science at Stanford. And then she returned to UCLA, where she directs a brain connectivity and cognition lab at the Center for Cognitive Neuroscience Analysis at the Semel Institute for Neuroscience and Human Behavior. She has her research combines functional and structural neuroimaging to examine the organization of large-scale brain networks, supporting development of social cognition and executive function. She has some amazing work on autism and like how insula and salience network is impacted in autism. She has been a pioneer in that field for a long time. And she's currently worked on understanding dynamic brain network interactions, which underlie cognitive inflexibility in, again, or disorders like autism. And she has published extensively in amazing journals. And I'm pretty sure during this course, they have read about your work. So I'd like you to take it away.
Thank you, Vaibhav. Thanks for the opportunity to present to this group. It was really a pleasure meeting at Washington University. We had some really nice conversations about a topic I'm not as familiar with. We were talking about mindfulness, and a lot of interesting things came out of that conference, especially about default mode network. So, yeah, looking forward to continuing those conversations as well. I think I have to share the screen, so I have. Okay, let me try that now. Okay, I'll do that and I will hit this button. So tell me if you can see now the whole screen.
Yes, yes.
Okay, perfect. Thanks. So, yeah, I wanted to talk about something a little bit different today. I actually gave a similar lecture at OHBM last year, but I'll give a little update for those of you who were not at that conference. And this is about a more big picture question in the field of cognitive neuroscience and network neuroscience. So, it's the idea that we could try to move towards a universal taxonomy of functional brain networks. So, I'll tell you about how that idea came to be. But first, we're going to already do a pop quiz. So this is the first question on the quiz. If you were watching, you can put the answer in the chat, whatever you think you have the answer. So the first question is, if you could, please name this network. You can put any answer you'd like in the chat. I'm not going to do any grading. That's not my role. But just I'm curious, if you had to give a name for this network, what would you call it?
I'll probably take a note, whoever is answering rightly.
And actually now, because I'm screen sharing, I can't see the chat. Vaibhav, would you mind reading some of the
Yes.
things that are written in
So
the chat? Yeah.
DMN with a question mark, and just two as of now. I think other people might respond. Anyone else?
Okay, well, let me give another, we can do another trial. How about this one? Please name this network.
Maybe people can unmute themselves and speak if.
Oh yeah, you can also talk or you can put in the chat. You can make a guess.
Is this the DMN?
Yeah, very good. Okay, this is one very good guess. Okay, we'll try one more, or I think I have two more. What about this one? Maybe tricky. I'll try one more. This is another one, last one. Yeah, it's funny because this is exactly what happens when you give this talk and you ask an audience, they don't know what to say. So it's not just this, that it's tricky. It's that we don't know exactly because we don't have a good definition of all of these networks, even though we talk about them all the time in the literature. So this is why I wanted to play this game, just to show you that we have, as a field, we haven't even decided what are the names of these systems that we're talking about. We have some ideas, but nobody can agree. And so that's part of the problem that I wanted to talk about today in the field. So as you know, about 30 years ago, Bharat Biswal discovered spontaneous activity is structured in this coherent way where you can understand the organization of large-scale functional brain networks, even when the participant is not doing anything in the scanner. So this is where resting state fMRI, resting state functional connectivity came from. And the idea, which was surprising at the time, is that in a low frequency range, you can see networks that normally are involved in attention and in memory and language, vision. All of these things are circulating in the resting brain at a very low frequency. And it's a very structured spontaneous activity. So it's not completely random. You can see the same networks we would see in tasks. We can often find them in the resting state. Oops, slides are going the wrong way. So the history of this field is now about 30 years old. And we just did a review on this to give some ideas about where the ideas came from and what are the major milestones in terms of thinking about the default mode hypothesis of the brain, which is now over 25 years old, this idea, and different network analyses and different data sets that are now being used to answer questions in network neuroscience, like Human Connectome Project and others that you probably know about. And people are using, intrinsic connectivity to look at even the fetal brain before birth. There's a lot of now ideas about how we should analyze data and best practices. And there's many, many data sets that are now available for people to use all over the world to, publicly available data sets that were made available through these open science initiatives and projects. So a long, long history, essentially, to tell you about what I wanted to get to today, which is this problem of what do we call the networks that we see in the brain. And in other fields in science, right, you have taxonomies for naming and describing and classifying. So with plants and animals, and you have all of these classification systems. And the whole point is so that we can talk between different labs and we can communicate effectively, and the science literature can build on itself. Obviously, we don't have this yet in our field. We talked about some of these taxonomy issues in my lab when Taylor Bolt was a grad student with us, and he did some meta-analytic work to find that across many, many different task fMRI studies, you can find that there's a general factor that explains over 50% of the variance in the task activation maps, and that factor looks something like the default mode, which is in green on the right in the Fox paper, and the dorsal attention, which is in yellow. Which explains most of the variance that you can see in task fMRI. And there's subtypes within these, general factors. This was a bifactor model to kind of explore the structure of cognitive neuroscience studies. And there's other subtypes that explain another 30% of that variance. So it's there's not that many different brain configurations overall, but what is really different is the way that we talk about them. So sometimes we use these terms like multiple demand system or executive control network. And in different studies, almost the same brain areas are being described by different names, right? So you can see some of these activations are highly overlapping, but one paper calls it the domain general system, another calls it multiple demand system, and another calls it the extrinsic mode system. So I think this is basically what happens when you don't have an overall taxonomy, you don't have an overall structure for naming things. Everybody names it whatever they like, right? This is what ends up happening. The default mode network was one that was more consistent. I think most of you are, when you saw it, you thought, okay, that looks like posterior cingulate and medial prefrontal cortex. That one has been more, systematically, I think, investigated. The problem, I think, is often in the other networks. The whole brain is, made up of networks, and we haven't really converged on how we want to talk about them as a field. So what's the problem? What's in the name, right? So there's one network that sometimes is referred to as cingulo-opercular. In some of these studies, it includes anterior prefrontal, anterior insula, dorsal anterior cingulate thalamus. In other studies, an anterior insular network is that I've called, Bill Seeley and colleagues have called the salience network, also includes dorsal anterior cingulate and and insula and subcortical and limbic structures. But then you have also other networks, like, for example, ventral attention network from Corbetta and colleagues, that's said to include temporoparietal junction, ventral frontal, middle frontal gyrus, and also including the insula. So to me, this presents a little bit of a problem for the field. Like, are we talking about three different systems that all share the insula node? Are we talking about different dynamic instantiations of the same system? What exactly are we talking about? These names came from three different labs that study different kinds of cognitive paradigms. And so they gave the names however they wanted to give the names based on the ways that they described the networks initially. But now, if we want to talk to each other across the different labs, we have to have more of a common language, right, in order to make some progress in the field. And so this is, to me, if you take one take home from the whole talk, is that we have to make progress on these functional brain network atlases in terms of understanding what we mean when we say this network. So if you are a person who's interested in studying the salience network, like myself, you might look across 4 different atlases and see, actually, in the Gordon atlas, the salience network looks like this. In the Yo atlas, it's actually combined with ventral attention network. It looks like this. In the Shire atlas, it looks like what you see in the bottom left. And in the Shen atlas, it looks even different. So, I mean, you guys can see without doing any math, right, that these are not the same. Even though they're calling themselves the salience network, they look different on the brain. The parcellations, the spatial topographies are giving them different anatomical locations. And so, to me, this represents the biggest problem for us as the field. Which salience network are we talking about, right? And this is just one illustration, but the problem is pretty obvious across different systems. And I'm a person who cares about the salience networks. I wrote a book on it. It's like part of my whole career is like focusing on this. And if I look at this and I say, and you ask me which one is the real salience network, I can't tell you because there's no right answer in this question. It's like, well, it's whatever the atlas says it is, and it's not a very satisfying answer because each atlas is calling it something different. So it just speaks to, it's almost like a philosophical problem we have in the field, that we keep coming up with names and they're not assigned in a systematic way to talk about one part of the brain. So maybe some people are not very bothered by this, but for me, like, I can't sleep at night thinking about, like, which one is it, So this is why I've been working on with the Organization for Human Brain Mapping to try to resolve some of these issues in the field. And particularly for us, like you saw the talk from Vinod Menon, we've been thinking for many years about this special role of the salience network. It might be involved, we think, in switching between internally directed actions of the default mode network and the externally directed, executive control or frontoparietal networks. Again, the names are varied across the different studies, so it's hard to keep track. But, we have this kind of model of large-scale neurocognitive network dynamics that we've been working with for 10, 15 years now. And I think a lot of people in psychiatry use it, as, Vaibhav is, talking about the review we did together. A lot of people use it to think about transdiagnostic, symptoms across disorders. And it's been an interesting framework, I think, for child and adult psychiatry. But in particular, the insula is sort of my favorite region of all of this. It's involved in so many things, like, affect, emotion, empathy, and pain are always describing insula activation. You have subjective awareness, interoception, somatosensory processes, also very much insular processes, and even the primary taste gustatory cortex is in the insula, so the taste sensation resides there too. Then you have high-level cognitive processes like conflict detection, switching, attention and inhibition. All the time when you have tasks that are tapping executive functions, you almost always see the anterior insula in particular active. So it's all over there. It's in everything we do, pretty much. And there's some nice work, I think, from Thomas Yo a while ago, where he was talking about this kind of idea of functional specialization and flexibility. So which brain regions are involved in a lot of processes and which are more specialized. And he looked again in a meta-analytic fashion with Bayesian hierarchical clustering, I believe. And it was looking across many different tasks, different experiment types, different cognitive processes, and which brain voxels are activated by which of these processes. And in this kind of meta-analytic way, you could see that the red brain areas are those that are involved in the most highly involved in different kinds of cognitive components. And again, you see the anterior insula there popping up, anterior cingulate, very much parts of the brain that are active across many, many different tasks. And so it's a functionally flexible brain region. We've done a lot of work on the insula in also different kind of meta-analytic connectivity modeling studies. And it's basically a very diverse brain region. There's a different way of looking at what you saw in the last study, but across all of these different cognitive areas or domains, there's a common functional fingerprint that all of the insula subdivisions seem to be involved in, to some extent in almost every task that is out there. It's the most highly diverse brain region that we could find. And this was some work done more than 10 years ago now, again, using meta-analytic connectivity approaches on task fMRI data. So oops, recording is okay. All right. So basically, we're asked the question, are these different parts of the insula communicating with the rest of the brain in different ways over time? And so the long story short is we found five different insula connectivity states. And in some of the states, like, for example, the one that occurs most frequently, state 3, many of the insular subdivisions have similar patterns of functional connectivity with other brain regions, whereas some states like state 2, which don't occur very often, there's actually more divergent patterns of insular connectivity depending on which subdivision you're looking at. And I think what was most interesting was this dorsal anterior insula. That's the one that's in red on the diagram, the red subdivision. The anterior insula is the one that's the most dynamic in terms of its diversity of functional coupling. So in different connectivity states, it's talking to more different brain regions over time. So we've really been interested not just in what's the insula doing, but what are different parts of the insula doing, and how can that explain some of its functional flexibility and functional diversity. So we've done stuff like looking at the dynamics of the insula ACC salience network across the lifespan. We had an undergrad in the lab who looked at between the ages of 6 and 85 using the Nathan Klein Institute data set, publicly available data set. There's some states in which the salience network is talking to the rest of the brain in terms of functional coupling, and other states where it's more isolated. And just, one example here is like state 4, for example, showed I guess I didn't put the slide in here, but basically showed a pattern where you see that more often in early life and in late life and less in midlife. So there's kind of different patterns of salience network connectivity with the whole brain that change as a function of a lifespan stage. And we've taken this to, into neurodevelopmental disorders like autism, which is sort of our favorite thing to study. And if you look at the insula and salience network dynamics there's you can use things like coactivation pattern analysis, a different way of looking at insula dynamics, and find that there's some patterns in the brain that involve insula, default mode, and central executive that occur less frequently in kids who are diagnosed with autism than in kids who are typically developing. And the reason I'm rushing through all this is just because I'm just giving some examples of why I focus a lot on the salience network and trying to understand what exactly it is, because there's so many ways that it seems to be involved in cognition across the lifespan and in atypical development, in particular in autism. It just seems like a relevant place to look in the brain when you're studying psychopathology. And even, I don't know if they meant to put this in here on purpose, but if you look at this paper from Insel and Cuthbert, they were talking about the research domain criteria and how we should, move away from symptom-based categories and move more towards integrating data from genetics, brain activity, and physiology to come up with more data-driven clusters of people that might be better served by specific interventions or treatments. And so my personal perspective is that we can probably get a lot of information just from the insular cortex about biomarkers that might be interesting for precision medicine, because there's so much information in the insula structure and function and connectivity that you can see is relevant in disorders that come on across the lifespan. So insula, again, my favorite brain region, and that's why I've become really into this question of like, what exactly is the salience network? What are we talking about? You can see across different mental illnesses. I can't remember how many different illnesses were included in this meta-analysis, but this is a voxel-based morphometry VBM study just of brain structure. And you can see again, across these many disorders, there was reduced gray matter volume, insula, and ACC, which, by many people's standards would be considered part of the salience network. I don't think they use the term salience network in the paper at all, but nevertheless, this is what you see across different across different mental illnesses. This one was one of my favorites that came from Chuck Lynch. He was actually working in the lab with Vinod Menon and myself back in the day, and then he went on and started his own lab, of course, and published this really great paper in Nature last year, 2 years ago, about the salience network. It actually seems to be bigger. It's expanded in individuals with depression. I thought that was a really interesting application. And they even used the data from the adolescent brain cognitive development study to show that kids who are 9 and 10 who later go on to develop depression, they also have an expanded salience network. So not only for adults who are already diagnosed, but even for kids, you can use the salience network to predict later depression. So this, to me, just shows how much relevance there is in understanding the signals in this brain area. The focus here was depression, but my suspicion is that you would find something similar across a few different disorders if you had the data to map this. And this is another area of interest. I was not very much aware of the addiction literature, but now I've become more involved in it as part of the ABCD study. And here, this is a paper showing a lesion network mapping of places in the brain where you get reduction in nicotine addiction and alcoholism for people who basically are showing lesions in this, what they're calling an addiction map. They call this addiction map a common brain circuit that's involved in all kinds of different addiction. But as you can see, it clearly involves the anterior cingulate and anterior insula, just one big salience network map. Again, I don't think they used the term salience network in this paper. So to me, it's terribly frustrating when you look across the literature and you keep seeing the salience network, and in some places they're not even calling it by the name, in some places they're calling it something different. And I wouldn't mind so much, except that when people are reading the literature, like yourselves in a class reading the literature, you wouldn't even see these links because they are not using the terminology that you're looking for. In fact, if you go into PubMed and you type in salience network, some of these papers won't come up because they didn't use that term. Either they use some other term or they didn't think about it in this network way. So I think it's very hard for the field to move forward until we can resolve some of this naming issue. And so I've been kind of obsessed now for 4 or 5 years with this question of like, how is a functional brain network defined? And how can standardization of network nomenclature impact clinical translation? The bigger picture question. And so I was at this alpine brain imaging meeting. I was not a good skier. I took one ski lesson and then I just sat by the fire the whole time. But during this meeting, they asked us to write a paper for a special issue. And that's when we first wrote this paper with Nathan Sprang. And we asked Thomas Yeo to come in because we always use his atlases anyway. So we thought, okay, let's ask him to be a part of this. And we tried to come up with some ideas for how to overcome the issue that everybody is using different names and they're not corresponding to each other for when they're talking about networks. So the first thing we did was say, okay, maybe we can propose an anatomical naming scheme where everybody just calls things by anatomy and they're no longer using terms like salience or whatever. They're just talking about the parts of the brain that are involved. So we thought, okay, let's just propose, we'll just have occipital networks. Instead of vision network, we'll say these are parts of the brain that are in the back of the head, and that's it. It's just occipital. It doesn't matter what it does, but we can go by a name. Everybody agrees on anatomy, even if you don't agree on function. And then we thought maybe you can do the same thing for the somatosensory. Instead of calling it sensory, we can call it pericentral because it's around the central cortex. And most of these atlases have something to do with motor, networks. Every atlas has something like that. Maybe it's broken down further. Then almost all the time you see dorsal attention network in the literature, but we said, why not just call it dorsal frontoparietal network? Because it could be attention, it could be something else, it doesn't matter, just call it frontoparietal. And here it's actually looks pretty good. It's in green in the Gordon and in the Yo atlas. There's some correspondence there, so that's good. And then a lateral frontoparietal network, sometimes it's called executive control network, sometimes it's called multiple demand network. This has many, many names. And so we thought maybe we should get away from the function again and just stick with the structural name. And then this was the one that used to be the salience network or mid-singular solar network or ventral attention network, the most confusing network of all. And so we proposed, let's just call it singulo-insular because those are the brain regions that are common. Even if you look at the Gordon, he actually calls these 3 different networks. So, we can ask the question, are they the same? Are they different? But in Yo's parcellation, they tend to go together. So, I think a lot of that is just subjective. And so we just have to make some decisions. And then this is the default mode network. But if we really just gave it an anatomical name, it could be called the medial frontoparietal network. And, this one seems very common and consistent, and people seem to agree on it, essentially. And so this was the preliminary idea that, okay, we could have at least 6, maybe more systems. And instead of calling them by their old names, we could just switch to an anatomical scheme. And hopefully the field would be more streamlined as a result. So that was very ambitious of a project. 2019, nobody was doing it. So it didn't work, I think, for a lot of reasons. Nobody wants to change their network names. Nobody wants to, do everything differently now after 10, 20 years. And so, people are also thinking about dynamics. What about the, moving between, nodes that change allegiance? What about the subcortical brain regions? We didn't even talk about them. What about inter-individual variability? Like the spatial location of some networks might not overlap completely in some individuals. And what about this, idea of fractionation? Sometimes the default mode can be 2 or 3 subsystems, anterior, posterior, medial temporal lobe, who knows, right? So what about the, these kinds of bigger issues about the hierarchy and scale? So we didn't succeed, basically. The first point, we didn't make any progress in this first attempt. So much of that you could predict. The reasons why we didn't make progress was because we know even before neuroimaging that cognition is served by multiple networks, and then cognition can be complex behavior can be served at the level of multifocal neural systems. It's not sort of one-to-one mapping between brain region and function, and also it's not one-to-one mapping between brain network and function. We already sort of knew that. And then there's this idea of neural context, that the functional relevance of a brain area depends on the status of other connected brain areas. So there might be, some reason that a set of nodes would behave differently in condition A than condition B because of what's going on in the network around it. And finally, the part about dynamics of understanding interactions between brain regions as they unfold over time. We don't capture that when we talk about a static parcellation. We're just saying, here's a snapshot of the brain. We're not really capturing what's changing. And, we know, like, this is some work from Taylor Bolt in our lab again, that a lot of these dynamics can capture a lot of things that they look like different patterns, but they're actually just like different time frames of the same pattern. So in this study, we found that 3 spatiotemporal patterns constitute almost everything else we see, which is like the task positive, task negative, and functional gradients and all of that stuff. So all of these things can be manifestations of these 3 main spatiotemporal patterns, which he modeled in this paper using a complex principal component analysis. So, we're not taking into consideration all these complexities when we make these simple naming schemes. But what we did was then, we formed this work group called the Workgroup for Harmonized Taxonomy of Networks. And Anastasia Yandiki was the person who came up with this very nice, clever name, WHATNET. And it was formed similar to the best practices on data analysis and sharing, COBITUS, which was an OHBM group that formed many years ago. Then there was a best practices in MEG and EEG. And so we formed this best practice group modeled after those groups, which is you get together with 20 or 25 people and you try to come with consensus on some of these issues. And so we wrote this paper, I guess it was 2023, and this was after 3 years of meeting on Zoom every month for, during the pandemic with 25, 26 authors and having discussions. Even there was a philosopher in the group, and a bunch of these people are those who created the atlases with cognitive neuroscientists like Randy McIntosh and Russ Poldrack and all of the people who you've read, their papers for 20, 30 years. We said, if we put them together and say, can we agree on some names, maybe we can come up with some consensus. Again, we failed. Again, like this, I'm telling you how much work we put into this, and still, like, we could not come up with some like nomenclature which everybody agrees on. These people are very stubborn, and nobody wants to change what they've been doing in their literature. So we wrote a paper about the controversies and the progress towards this, and I think the paper is like very, very long, but it lays out a lot of the conceptual issues, methodological issues, and where we think the field needs to go. And when we did the paper, we did the same thing which I showed you at the beginning of the talk. We went on Twitter and asked people to name the network, and wanted to see, like, hundreds of people across the internet what they called the different networks. And some of them were actually named very easily, like visual, motor, and default mode. People were in agreement. But if you ask about like salience, frontoparietal, and those ones, nobody agreed like what to call them. So they gave completely different names, random things. So that was part of the controversy. The progress was, we thought, okay, obviously nobody will agree on these names. So instead we gave some general guidelines, like if you're looking at a task, it's not necessarily the same as looking at like a network derived from REST. And so try to avoid giving some like functional idiosyncratic name to the network and try to match it to some atlas, but then again, the atlases are showing different things, so that's not always going to work. And so, we gave these as like general guidelines, try to talk more about the anatomy. And so we got this as basically an OHBM endorsed best practices thing. And we thought, at least we put some guidelines. Let's see if people use them. I don't think it was widely used yet, or I don't know if too many people are following this controversy. Maybe it's just me. So finally, we came to the conclusion that we have to give people a tool, right? We have to provide a way to make it easy to do this, what we're recommending, right? Because if you're the grad student, right, you come into the lab and you show your advisor, like, here's my data, here it is. Now, what should I do? I found some nice activation. I want to make a story and write a paper and get a publication, move on with my life, right? So in the beginning, if you came to my lab, I would say obviously it's a salience network. You can see there's Insula and ACC and all of this stuff. You can write a whole story about that based on the salience network literature. But if you go to Thomas's lab, he'll say, oh, it looks like it overlaps with the ventral attention, whatever. And so you might look at that ventral attention literature instead and write a whole story in that way. And if you went to WashU, you would maybe start with cingulo-opercular network, and then recently they've renamed it to action mode network. And then basically you would have a whole different story about this activation map. Their map hasn't changed, but the way you interpret it has changed based on which lab you grew up in. And to me, I don't think that makes any sense. Like we shouldn't be giving different interpretations just based on who is your advisor. It's not a good way to build on findings in the field. So that's what's always been bothering me, is that like every paper you read is talking about things from the perspective of that particular lab that you grew up in. And so this is what we did. We came up with this Network Correspondence Toolbox. So Ruby Kong, she was working in Thomas Yo's lab at the time, and came up with this way of letting you sort of compare across all the different atlases to your own findings, to make the interpretation a little bit easier. Even if you don't agree with the labels, you can say, well, this is the label given by this atlas, and this is another label given by another atlas. So what we did was essentially, what Ruby did was is very simple. It's just computing dice coefficients between the spatial maps that you're inputting, taking your data, whatever it is, comparing it across all the atlases. I think we have like 16 or 20 atlases in the toolbox, and we're always adding new ones. And it computes the dice overlap between your map and every other network in every atlas, right? So it can give you some output that says like how much overlap there is with Gordon's, dorsal attention versus Yo's, default mode. Like it gives you all this quantification. And the way it looks, and what I like the best about it is that you can compare different atlases just for fun. So this is a Python toolbox. You can download, install it, and hopefully it's easy to use. So the idea is like if you let's take two atlases, Yo 17, Gordon 17, and compare them to each other and say which networks in Yo overlap with which networks in Gordon. And you can see like just from this simple comparison that what is called salience ventral attention B in the Yo network is called cingulo-opercular and salience in the Gordon network. So they're the same. The spatial overlap is very high, but they have different names. I don't know why people are pushing against this idea that they're different. If they overlap like 90%, then they're probably the same. And so like you can talk about it as much as you want, but like this is a way to quantify, what's actually happening in those atlases. And you can take your own data. So on the top is like a yellow map of some, let's say working memory task. And you say, I want to compare this across 4 different atlases. And that's what this toolbox will do for you. So it'll output this dice overlap. The sort of network clock is how much overlap there is with your findings and all of the different networks in those 4 atlases. And then on the right, it's just a nice map. Oh yeah, is there a question? Please, yeah, go ahead.
Hi, yeah. My question is that if there is such considerable overlap between brain regions and how they, between brain regions, why do we have separate atlases in the first place? Like what exact function are they achieving?
Yeah. So do we have so many different atlases? That's the question.
Yeah, and yeah, that's the question.
Yeah, no, it's a great question. It just happened that different labs across the world decided to make different atlases. They all use like slightly different algorithms for parcellation. So that's why, they all think their parcellation is the best one, right? Because they have developed some way of dividing up the brain, and they think like mine is better than that one, and this is better than that. So you have, 10, 20, now probably 30 or more different atlases because they came from different groups with different methodological assumptions, and these are all using resting state fMRI data, but they use different criteria for delineating the parcels, right? Slightly different, ways of doing it essentially. And though many of the labs, they're mostly like oftentimes engineers and computer scientists. Sometimes there's neuroscientists, sometimes there's cognitive, psychologists and things. But I think when you approach the problem from an engineering standpoint, you don't necessarily care that it doesn't overlap with somebody else's atlas. You just are, doing your own method and trying to show like this is the best way to parcellate the brain. So I think the motivation wasn't there to like, let's make it consistent across other groups. The motivation has to come from us who are like the brain people and say, actually, for the field, it would be better if we had some consistent overlap. But I think, all of these things just originated independently in different research groups at different times. So now I'm trying to make them talk to each other after the fact. Is very difficult. People don't want to change their ways. So yeah, essentially what we did was just create these things. So, like this is a close-up of that working memory contrast. But if you put this in your paper and you can say, well, my finding overlaps with Gordon's dorsal attention frontoparietal. It also overlaps with, Yo's salience and control A, B. So even though they're different names, you can make it easier for the reader to compare if they would like to do that, and easier for yourself too. And then sometimes you have like really different maps that are like an ICA component, and you just want to give some label to it. Is very difficult because it overlaps with a lot of the brain. But with these maps, you can at least say, well, here's how much overlap there is with each of these systems. You don't have to use it in this way. These are just examples. And so this was written up as a kind of a news article in the transmitter. They called it the decoder tool for translating neuroimaging results, which I sort of like that idea that you can use it as a way to translate the findings from your own study to other studies. Yeah, and this was yesterday, or when was this? Last summer, when I was giving the OHBM keynote, they asked me to talk about why standardization matters, and this author in the student postdoc special interest group did a really nice review of our study, talked about what the problem is with the variability in network definitions, and how you can do quantitative comparisons with the brain network with the Network Correspondence Toolbox, and the fact that we put in some stats in there if you want to do comparisons between different spatial maps, and you can look across atlases for improving your sort of definitions, and it should be user friendly. So if you do use it and have problems, let us know. And the idea is to move more towards the standardization, more towards the universal taxonomy, which I think it hasn't been reached yet, but that's the goal. And so I think as we get further and further down this path, 30 years have gone by, who knows what we'll look like in 30 years. But as we move further along, I think it's going to be increasingly important to try to arrive at some consensus about these network nomenclature issues, the naming, what do we call them? And the Network Correspondence Toolbox is, I think in the meantime, it's a tool to help us translate findings across labs and across different atlases as we work towards this more universal goal, which I'm not sure if we'll be able to achieve in the long run. And this was the group, the sort of WHATNET group. We had many folks working on it over the years. This was the OHBM meeting in Glasgow where everybody got COVID. So that was the bad news there. But we did make some progress on the toolbox. And, we can always add new atlases. So there's, you don't just have to use resting state fMRI parcellations. You could use histology-based or, site architectonics kinds of atlases. If you want to, you can upload them into the toolbox, and we can, make that a function. And there's, there's so many different atlases, right? So, they all have these different properties. So we're trying to make it so that whatever atlas you want to use, you can compare to some other atlas and compare to your own findings. And we're also thinking about, all these atlases are usually based on adult brains from 35 to 45. They're kind of very standard adult neurotypical brains. But there's all these changes in networks with development. So in early age, you often see things, not as well formed in terms of the network structure and topography. So we're working on networks that are like adolescent specific, for example, working on like this is Gail Doucet and Vince Calhoun's group. They do a lot of work using independent component analysis to define networks. And we're trying to figure out, if there should be specific ones for specific age ranges. And I have been meaning to write a grant to try to come up with like a lifespan atlas that would be useful for some lifespan and dementia and all those studies. I haven't gotten around to it yet. And NIH isn't really working anyway. So this is the sort of end goal, is like trying to move towards a universal taxonomy of functional brain networks, where we could sort of have some agreement upon what we're calling these systems in the future, if not right now. I don't think we're there yet. I think that's the tricky part. Like it might take some time for the field to accept some of these issues and accept some of these proposals, but we're trying to work on it very much. This is my lab at UCLA and my collaboration network on the right. And these are all the folks who are part of the WHATNET and some of the funding over the years that have contributed to what I've talked about today, and the big data sets and the people providing code. It's really a group effort. So yeah, I'm happy to take some questions if we have a little bit of time for discussion. So I think that maybe now I can see the chat as well. I didn't see it before. Yeah. So, and if you like, you can maybe read out your question. I see somebody wrote in the chat.
Yeah, Prakriti, you had a question. Yeah. Would you like to ask it directly?
Okay.
Yeah, okay. We can just maybe I'll read it for everyone.
Okay.
Yeah, thanks for the talk. Since the salience network seems to be involved in a litany of things, including multiple developmental and neurodegenerative disorders like FTD, how do we go about its utilization as a biomarker? Can we perhaps build a cognitive profile for it? And can behaviorally measure damage or degeneration in it? Or is it that the detection of impairment in the network merely limited to discovery from the imaging techniques?
That's really a great question. Thanks for bringing that up. Actually, so I think we often use this term biomarker, but I don't think we use it accurately, because if it's something that's a true biomarker, would be sensitive and specific for that disease. So if salience network connectivity was a biomarker for FTD, then it would tell you that this person has FTD, but they don't have a different dementia, right? It would be specific. But I think, as I've alluded to, you often see salience network dysfunction. You see it in autism, or you see it in anxiety, or you see it in depression, or whatever. So if it's often disrupted, it's not a specific biomarker for that disorder. It's more of just a marker of general brain health, or something is going wrong, or it's more like a general dysfunction indicator. So I think that when we use the term biomarker, we're just being too optimistic. Like it's not really functioning that way. But I think the question that you're asking about, like, basically, like, it perhaps build a cognitive profile and all of that. I think that's part of it. Like, I think, like, you can use brain metrics, but there's also all these other measures. That's what the sort of research domain criteria suggests, that maybe we have like genetic risk factors combined with lifestyle, health measures combined with the brain measures, that together can help predict future onset of neurodegeneration. But yeah, I think you wouldn't necessarily only want to rely on imaging techniques for this. In fact, you'd want to incorporate all the other information that's relevant. So I think we are kind of kidding ourselves when we say like biomarker from neuroimaging. I think that's too optimistic at this point.
Yeah. I think there's one suggestion. Okay. Can you direct me to some papers discussing change in resting state connectivity in stroke patients? Maybe some groups are predominantly working in that space. Do you know who working on stroke patients and resting state connectivity?
Stroke, let's see. Yeah, there's many groups working on this. I don't work on it myself, so off the top of my head, I think I don't know. But like, there's a lot of the neurologists. I'm thinking of Mike Fox, but he does more like TMS treatment for depression and things like that. But a lot of the neurologists, like, who treat stroke patients, are now using this idea of lesion network mapping. Like, they have a lesion to some brain that's due to stroke, and that affects some large scale network, and that it affects the function. You can almost like predict what's going to be affected because of the sort of network structure. So yeah, I'll have to think. It'll take me a minute to come up with the names of exactly who's working on this. But if you do a PubMed search with like stroke and default mode or something, you will definitely find a lot of this literature. A lot of it is out there now.
There's another question. Can these cortical networks also use subcortical areas? It does seem intuitive to check whether the insula is also connected to the limbic system along with the ACC. There are also certain atlas do not measure these subcortical areas. How do you resolve that within your toolbox?
Yeah, exactly. So we don't resolve it in the toolbox. We just use whatever atlases are available. There's a couple of them that do include the subcortical regions. I believe there's a brainotome atlas that includes subcortical regions, and there's one from Michael Cole's group that does include subcortical areas. But the reason historically a lot of people didn't include it was just because they were using surface-based, like cortex-based parcellation approaches, and doing everything at the surface, and sort of ignoring the subcortical areas in the parcellation schemes, and that way ignoring the subcortical areas in their atlases. And there's not a good reason for it. They just were using approaches that didn't capture the subcortical regions. But I think it's very important to capture them, because things like the thalamus, amygdala, cerebellum, they're all like part of these large scale networks. So we can't, the basal ganglia, we can't ignore them, but we sort of, we sweep them under the rug sometimes when we talk about the whole brain, even though we know they're there and we know they're important to the functioning of these systems. So I think the truth is we have to start incorporating them more fully into our models. And there's a couple atlases that do that, but many of them still don't.
Okay. So thank you for such a wonderful talk, for addressing such a pertinent issue. Hypothetically speaking, if we didn't have any atlases at all, how could we go about creating a standardized one? Basically, what would the perfect standardized atlas look like?
Yeah, well, whoever made the atlases will say, I already made the perfect one. It's the one I made, So everybody, that's why we have so many, because everybody looks at the atlas and says, actually, I can make a better one. And then they go into their lab and they come up with a new way of parceling the brain. And that's part of the problem. Then you have like more new atlases. It's contributing to the issue, right? Because they think, well, my algorithm is better. I did a better job, or I included the subcortical areas, so now mine is perfect. But I think the perfect standardized atlas to me would actually be useful across the lifespan, because right now it's based on the typical perfect adult brain. So not every a brain is going to be like that. If you have stroke, you have dementia, you start to see degeneration. If you have early life development, some of these networks haven't, large long range connections haven't even formed yet, right? So to me, the perfect atlas would be flexible enough to incorporate different ages and different pathologies and still let you compare them to each other. So part of what I was writing a grant with Thomas Yeo and Nathan Spreng to do a lifespan atlas where we talk like start from young ages and go all the way up to, the late life. And I'm still trying to, and actually Thomas Yeo has some good methods in the lab to incorporate these like individual variations. So I think we're gonna do it eventually, but I got sidetracked with all the like drama in the United States and things happening with funding here. So I haven't done it yet, but we'll make the perfect atlas eventually that includes the whole lifespan.
I think, any other questions from the students? Okay, I have a couple. So do you think like all the cortical area or even like the subcortical or the cerebellar areas would be involved in one or the other network? Could there be regions which are just like not in any network? They're just standalone areas. And if there are such areas, how can we like track them, or how can we like include them in these atlases?
Actually, I think like there's been a lot of work done on the cerebellum. There's a lot of folks who have looked at the cerebellum and connectivity to specific networks. I think Randy Buckner's group and some others. So the cerebellum is pretty nicely mapped in terms of how it talks to other cortical networks. And some of the other brain regions, I think they haven't been as closely. I think the thalamus has been. I think Damien Fair did some of that thalamocortical, mapping. So there's a few, there's a few papers out there that have tried to map specifically the cortical subcortical connections. But I think you're right that they're not widely part of the big atlases that people always use. They're not necessarily in there. So I think they just have to become more popularized, or there has to be like more standardization in terms of incorporating that. Like what I was talking about with all these problems with the naming, like it's even worse if you try to then bring in the subcortical regions because people haven't spent as much time there as well. So I think maybe we're getting there. It's just slowly.
And what about like any new networks that are found? So like we had Heather Mikowski recently. She had this paper she was working on the pain network, which is separate from the insular network slightly. It's there, but it's always separate.
Oh, really?
So like how if more such networks come in, how could that be important?
Yeah, I mean, I think that's the problem. Like, it's an ill-formed question. It's not, we don't have a set number of networks, right? Like I think if there is a pain network that's separate from the salience network, I'm sure there's times when it overlaps in terms of the dynamic function of it. Like there might be, let's say 50% of the time it overlaps and 50% of the time it diverges. And so that the dynamic aspect is never captured in these static networks. And we're using them as approximations because maybe like some majority of the time the brain looks this way, but there's so many deviations from that approximation that I think we're again, like we're imposing a network structure on a brain that's constantly fluctuating through, different configurations. So I think the best we can say is that we're using the parcellation schemes and the atlases as models or as something to help us understand the brain, but we're not fully capturing the dynamics. We're not fully capturing like and like I said, like we can actually explain everything that's going on in 3 spatiotemporal patterns. You don't even need like 7 or 8 networks, but the spatiotemporal stuff isn't captured in the static map, right? We use the static maps to help us interpret what we see. So I think we make a lot of simplifying assumptions as scientists because we have to make progress. But I don't think that any of the atlases are correct. I think they're just a best approximation that we can use to build on, but they're not sort of correct in the you know, gold standard kind of sense, because there is no answer to the question how many brain networks there are. The question, I mean, that is like sort of like depends on how many do you want to see. So if you do clustering analysis and you say, tell me what's the optimal number of clusters, if you look at Yo's atlas, like, you know, 7 networks was optimal, but so is 9. You know, is why are we using 7 instead of 9? Like they're both optimal. So it's that you guys know more than me. Like these are just like engineering and math problems, and we're just using them to help understand the brain. But there's no like right answer. We're just using the thing that helps us move the field forward the best.
Yeah, yeah. Great. Any other questions from the students? Okay. Awesome. Thank you. Thank you for this lovely talk. Thank you for giving us your time. And like, I hope you achieve your dream of getting a standardized atlas over the lifetime. Hopefully within our
Probably I'll retire before then, so one of you has to do that.
I think somebody in the audience will take it up definitely.
Thank you.
Again, if students have any questions, I think that if you're okay, they can reach out to you and
Yeah. You can email me. It's very easy to find me. So yeah, I'm very happy to take any questions. And yeah, thanks. It's very nice. Nice to chat with you all.
thank you. See you. Bye-bye.
All right. Bye.
See you at OHBM. Okay. Thank you, everyone. See you tomorrow.